What's up, Fifi Bio Penguins? So today we are going over Unit 5. And again, I keep losing my screen. So this one has to do with heredity. And so you're probably wondering, like, why are we doing this? Like, what is happening? Like, why do we even need to do... Ooh, wrong button. Sorry. I should have gotten the YouTube pull up in advance and I forgot to get pulled up. So why are y'all penguins? Y'all are penguins because of the fact that you have your nice little cute little tuxedo. You are dressed for success. And so like I always tell students penguins because of the fact that they are dressed for success and they are ready to rock the AP exam and they're ready to go with all of this. So like that's why I kind of call y'all penguins is because of the fact that y'all are dressed for success. Okay. So I got this pulled up. So now I don't have the chat. That's weird. Anyways, so daily review that we have. So on Instagram, I've got my daily review every single day. I post review on there for y'all. I do like content reviews, so I call them like the Insta-Review, but they're just kind of quick little reviews for you. There's a 374-page review guide that I have on my Weebly. So I go through each of the I can statements, and I have topic questions for you in each of those. And then I also have like multiple choice and fair responses for each of them to try to help you out with that. Okay, so then I've got FRQ Friday. So every single Friday, I've been recording videos for you to go through all the FRQs. I will be, I think in 2 weeks, I'll be done with all of the FRQs that have been released from 2013 to current. And then there's, of course, 120 review games on my website, as well as review PowerPoints. Plus, I do all these little reviews every other weekend for y'all. Someone says, you're a junior, will you be able to finish studying for the AP exam? You want to do the exam as a trial? I don't know. It depends on if you've studied for the exam or not. Okay, so today's plan, we're going to do meiosis. We talked about meiosis last weekend. We talked about mitosis. So we're going to talk about meiosis. We're going to talk about how mitosis and meiosis differs. I can't move my camera because of the fact that I can't find that screen on the Streamlabs thing. We're going to do genetics. We'll talk about Mendelian genetics, non-Mendelian genetics, as well as chromosomal genetics. And then we have some practice questions. And then I figure I can open it up for any type of Q&A. Okay. Y'all can write your questions into the chat, and as I go, I can answer them. I do find that there is a lag between when you say it and when I see it. It's about 30-second lag. So hopefully you can get your question in there, and then I'm hopefully still on the same slide. I can answer whatever question is you have. So first things first, we have meiosis, right? So the first step of meiosis is the same steps that we saw with mitosis. So we have prophase 1. So in prophase 1, we're going to see that the chromatin is going to condense. Our sister chromatids as well as our homologous pairs are going to line up. This is also when we see crossing over. So crossing over is going to be when these non-sister chromatids, so the sister chromatid from the two parental, so the from the father's chromosome as well as the mother's chromosome, maternal and paternal. Okay, so when we have those two chromatids, they overlap and they're going to exchange genetic information. So these outer ones that are all blue or all pink are going to be considered parental chromosomes, and the ones that are on the inside that have the overlapping that took place, the crossing over, that they exchange their genetic information, these are going to be considered recombinant chromosomes, okay, because of the fact that they are a combination of the two parents' information, okay. So after prophase, again, as I said before, prophase is when we're preparing to divide, okay? We then move into metaphase. Metaphase is going to be when the homologous chromosomes are going to line up on the metaphase plate. Now notice I put homologous chromosomes in like big letters, okay? So if a couple years ago, they had a FRQ question where they asked you the differences between mitosis and meiosis, and one of those differences was, of course, homologous chromosomes are in metaphase 1 versus sister chromatids are in metaphase of mitosis, okay? So don't just say it aligns on the metaphase plate. Like, what is it that's lining up on the metaphase plate? So be specific. So homologous chromosomes are going to line up on the metaphase plate. This is when we see independent assortment. The maternal and paternal chromosomes will line up independent of each other. So sometimes they'll be on this side and this side, and sometimes they'll be on this side, okay? So that allows for a little bit of variety that we see in the gametes, okay? So after metaphase, we move into, of course, anaphase 1. Anaphase 1 is when we're going to see the homologous chromosomes are going to move apart, okay? And so they're going to move into, of course, their two new nuclear envelopes. And so now at the end of meiosis 1, after cytokinesis, we're going to see that we have two cells, and these cells are going to be haploid cells, okay? So they're haploid cells, and they have already a duplicate. So you will not have to go through DNA replication in between meiosis 1 and meiosis 2 because you already have your duplicate chromatid. And so they're going to be identical, and they're going to be, of course, those two chromatids are identical, okay? Technically identical. Not really identical because of recombinant chromosome, whatever, okay? So they are already haploid. So then we move into meiosis 2. Okay, we again have the same steps. Prophase 2, our sister chromatids are going to line up. Our chromatin condenses. Metaphase 2, the sister chromatids align on the metaphase plate. Anaphase 2, our sister chromatids are going to move apart to the opposite poles. And then telophase, we have our nuclear envelope between our haploid nuclei, and then we would go through, of course, cytokinesis, which would then give us our 4 different cells. Okay. Now, knowing the difference between spermogenesis and oogenesis is not actually in the curriculum, so you don't really need to know the difference between what kind of gametes. We just need to make sure that we understand that this is how gametes are made. Also, keep in mind that meiosis 1 is going to deal with homologous chromosomes versus meiosis 2 is dealing with our sister chromatids. Okay. And so we need to make sure that we can understand the differences between mitosis and meiosis, okay? So mitosis is going to have a difference in the parent ploidy, right? So let's see. They both have the same diploid. So they're both going to start out being 2 end cells. The difference then, we each go through, again, 1 round of replication. And after that, we go through 1 or 2 rounds of nuclear division, okay? So here we start out with 2N, okay? We go through replication. So now we have, again, 2 sets of chromosomes, but there's going to be a duplicate. And then we go through 1 round of division, leading to, of course, 2 cells, versus here I have 4 cells. Those daughter cells are going to be diploid versus the daughter cells being haploid, okay? How does it compare to the parent cell? The cells are going to be identical in mitosis because of the fact that I went through this replication, and I equally divided those cells because of the way that I went through prophase, metaphase, and anaphase and telophase, okay? Versus meiosis, they're going to be just genetically distinct because of crossing over that occurred during prophase 1 and the independent assortment that occurred during metaphase 1. We're going to end up with genetically distinct gametes, okay? These cells are going to be different. Okay, crossing over does not occur in mitosis, but it does occur in prophase 1 of meiosis. Independent assortment does not occur in mitosis, but it does occur in metaphase 1, which my little penguin's on top of it. I don't think you can see the metaphase 1, but it does say metaphase 1. And so we need to make sure that if they ask us to compare and contrast, that we mention mitosis and we mention meiosis. So we can talk about what you found in both of those. Okay, so inheritance patterns. Okay, so we can have complete dominance. With complete dominance, the dominant trait is going to completely mask the recessive, okay? And the way that it masks it is because of the fact that that DNA is going to make a functional protein, while the recessive trait might not make a functional protein or might not make a protein at all. So we're able to see that dominant trait or that dominant characteristic, okay? So our homozygous dominant and our heterozygous will look the exact same because the dominant trait will mask the recessive, okay? Codominance is going to have to do with that you're going to see both of those independently of each other, okay? So with this, I can see both the blue and the yellow because you see both of those independent of one another, okay? And so with that, you're going to see that your heterozygous is going to have, of course, both traits. And then, of course, each of the homozyguses will have their own characteristics. Okay, and so this is also like AB blood type. So you have A blood type, you have B blood type, and if you have an individual with AB blood type, that means they have both the A glycolipid, and then the B glycolipid on there. Okay, and then incomplete dominance is going to be where you blend the two. Okay, so we've got our 2 different traits, and then the heterozygous of it is going to have both the 2 blended together. So if I blend blue and yellow, I get green. Okay, and so those are the differences we might see in terms of how things are inherited. Okay, so now students always ask questions about monohybrid versus dihybrid. Monohybrid means that it's heterozygous for 1 trait. Okay, having these ratios memorized does help you, but you are able to do a Punnett square on the exam and be able to work through those pretty quickly. Okay, so with complete dominance, we see a 3-to-1 ratio where we have 3 of them with that dominant trait and 1 of them with a recessive trait. Incomplete or codominance, we're going to see a 1 to 2 to 1 ratio where we have 1 that's going to be this dominant trait, 2 that are going to either be showing both traits with codominance or will be the blend with incomplete dominance. And then we have 1 that's going to show the other dominant trait. Okay. Now it's important for us to understand that dominance does not mean prevalent. Okay. So just because something's dominant doesn't mean that it's found in a large number of the population. Okay. So for example, having 5 fingers, polydactyly. This is a recessive trait, okay? So because of the fact that I have 5 fingers, I am homozygous recessive, okay? There is a family in Brazil that I saw a BBC video about in which the entire family, or most of the family, has an additional, I think it's somewhere like, it's like right here. This is their other finger. And they actually have 6 fingers and 6 toes on each appendage. And so that's actually a dominant trait. And so if one of them individuals with mated with somebody who only had 5 fingers, there would be potentially a 50% or 100% chance of the child having additional digits. Okay, so dihybrid is heterozygous, and so this means that it's heterozygous for, of course, 2 traits. That's where the di. And so we'll see with complete dominance a 9-to-3 to 3-to-1 ratio, and versus incomplete or codominance is a 6-to-3 to 3-to-2 to 1-to-1. Okay, and so you kind of see that 1-to-2-to-1 ratio here, and then you have a 1-to-2-to-1 ratio there. So you can kind of see the ratio. Again, you can do the Punnett square and calculate this. If I was to do a Punnett square, I would do 2 monohybrid crosses and then multiply them together. So I'm going to show that to you real fast. So if I was to see, okay, well, yellow. If I was to cross an individual that's heterozygous for the yellow trait, I would find that 3/4 of them would be yellow. These 3 are yellow. And then 1/4 would be green. Okay. If I was going to do the round versus wrinkled, I can see that 3/4 of them are round and 1/4 of them is wrinkled. Okay. So if I wanted to instead know how many of them are yellow and round, I'm going to multiply 3/4 by 1/4 because the probability of yellow is 3/4. Probability of round is 3/4. So if I multiply together, 3 times 3 is 9, 4 times 4 is 16. And so you could do that with each of these traits to figure out what yellow wrinkled is, see what green round is, or what green wrinkled is. And you would see that 9-to-3 to 3-to-1 ratio that I just mentioned over here. Okay. So you could calculate it if you don't actually have them memorized. Okay. So in terms of inheritance patterns, autosomal inheritance means that it's on one of the non-sex chromosomes. Okay, you have 23 pairs of chromosomes. 22 of them are autosomes. They just code for other things in your body. Okay, and then you have one set that is your sex chromosomes. I think they're also called an allosome to some people. And these are the ones that are going to determine sex. Okay, and so if you're XX, you are female. If you're XY, then you are male. Okay, and that's of course in humans. Okay, other animals have other ways that they determine the sex. And so, if, like for example, like chickens, they have a ZW system, and the male is ZZ and the female is ZW. So like it does vary in different populations. And there was a question a couple of years ago in which there was a certain type of fish, and the fish was also a ZZ-ZW system. And depending on temperature, it would develop as a male or a female. So there's a lot of differences that we might see here in terms of like the sex. Okay, sex linked means that it's going to be located on one of those sex chromosomes. So it'll be on the X chromosome, it could be on the Y chromosome, it could be on that Z or that W that I mentioned before. It just means that it is on that sex chromosome, that allosome. And then we have maternal inheritance. This means that it's in the DNA that's found in your mitochondria or your chloroplast. So according to the CED, it's important that you understand that the mitochondria is inherited by the mother. The chloroplast is inherited by the mother. So you're going to find that the pollen is going to be the male. And then you, of course, see that inside the pollen, you have that ovule. Inside the ovule is the egg, and that's the female, okay, in terms of a plant. And so there was a question, I believe, on last year's FRQ where they said that the male was, I think, green, and the female was like yellow spotted. There was a certain characteristic, and they wanted you to predict what the offspring will look like. And everybody kept doing Punnett squares, but it was important to understand that that was a mitochondrial trait or a chloroplast trait, can't remember which one. And it was inherited in that chloroplast, in the DNA that's found within that organelle. So this would be maternal inheritance. So we'll see that if the mother has the trait, all the offspring will have the trait. Or if the mother does not have the trait, none of the offspring will have that trait. And then we can also see information about linked genes. Okay, so linked genes means it's found on the same chromosome. So you'll inherit that trait together. Okay, so if we look here at this pedigree, we can see here that I have 2 parents that are unaffected, and I can see that there is an affected child. This tells me that it must be some type of it has to be some type of recessive, because you have 2 unaffected parents with an affected child. Okay. I also know that this is going to be autosomal because of the fact that you see an equal amount of males and females being affected by it. Okay, so this tells me that's autosomal recessive because of that. Okay, Also, in order for this female to be affected, that male would have had to be affected. And that male only has one X chromosome, so I would have seen him affected here. And so since this female that has two X chromosomes and it being recessive, she would have had to have two faulty X chromosomes, okay? In this one, I can see that I have two unaffected parents, and then I see an affected child, okay? And that tells me that it must be, sorry, I'm ahead of myself. So here I see that I've got a female, an affected male, okay? And so that tells me that this must be, again, some type of recessive, and it tells me that it must be sex-linked because of the fact that the male only has one X chromosome, this female has two X chromosomes. Those faulty X's then, of course, pass that on. And you can also look here. You can also see that there are way more males. There's 5 males versus 3 females that are affected. So that kind of helps you to see that that one would be X-linked recessive. Here I can see that every single parent has the trait, and so every single generation you see that it's affected. Here I've got 2 unaffected parents, and then the offspring is unaffected, so that tells me that it must be some type of dominant potentially because every generation has it. So autosomal dominant. And then here I can see that these ones, you have your female that is unaffected, and their children are unaffected. But if you look here, you got a female affected, 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 affected. So all the offspring are affected. Again, female, all affected, female, all affected. So that tells me that one is mitochondrial inheritance, maternal, chromosomal inheritance, whatever you want to say. Not chromosomal, sorry, chloroplastal inheritance. It has to be inherited from either the mitochondria or that chloroplast. So in terms of my chromosomal inheritance, it's important for us to understand about the crossing over and independent assortment. So crossing over has to do with, again, the non-sister chromatids are going to exchange their genetic information, which allows us to have recombinant chromosomes. So all of our gametes are going to be genetically distinct because just the chromosomes themselves are going to allow them to be different, okay? In this independent assortment has to do with that we see that they're going to independently align on metaphase plates. So our homologous chromosomes are going to line up on that metaphase plate. So the maternal and the paternal can be this way versus this way. And that happens for all 23 pairs of chromosomes. So it's independently how they align. Law of segregation states that we're going to see that we're going to have those homologous chromosomes will segregate and move to opposite poles. So if this chromosome was for, I don't know, the polydactyly gene, and this one was for non-polydactyly or the normal allele, then those would separate opposite from each other. So we would see these alleles segregate or moving to opposite sides. And then random fertilization, any sperm confused with any egg, which then leads to a variety in our offspring. Someone says how closely linked genes are on a chromosome. So you could find this by recombinant chromosome, like recombinant calculations. So you can calculate the number that are recombinant over the total, and that tells you the percent recombination, which tells you the distance. So if there's like 17% recombination, that means that there's 17 map units apart. If there's 45% recombination, that tells you that there are 45 map units. You'll never have a number that's larger than 50 because 50% treats it like independent assortment and that they're treated like they're on different chromosomes because so much crossing over takes place that they end up being on different chromosomes. So here we can kind of see that law of segregation. So law of segregation, we can see that those alleles are going to segregate from one another and they'll end up in different gametes. Versus here we see the law of independent assortment. They can independently align on metaphase plate. So here I have all the reds on one side and all the blues on the other. Here's one blue, one red, and then one red, one blue. So that can allow us to see a difference in our offspring. Okay, and so the chromosomal base of inheritance is taking about that. We're actually going to see that the traits are on these chromosomes, and the chromosomes then are passing down the trait to the offspring. Okay. Now, there's a lot of different genetic disorders. There's none of them that are in the official CED, but these are different ones that I just want to make sure that you are aware of. So, for example, sickle cell disease. This is usually brought up with malaria, and so if you are homozygous recessive, you're going to have sickle cell disease. If you're a heterozygous, you will have sickle cell trait, which means that you're not going to show all the symptoms of sickle cell. And then if you're homozygous dominant, you do not have the sickle cell disease at all. Now, in terms of malaria, malaria cannot affect sickle-celled red blood cells. And so if you have sickle cell disease, so you're homozygous recessive, then the malaria cannot affect your red blood cells. And then if you're heterozygous, you have a resistance, like you're not as likely to be affected by malaria. And so we're going to see that they could show a little crossover with that. Tay-Sachs is a autosomal dominant trait that is brought in. I'm sorry, I said that wrong. Tay-Sachs is homozygous is autosomal recessive. This is going to affect the proteins of the brain. They have non-functional lysosomes, so they can't break down fats, which builds up to something in their brain. So what the college work can do is they can connect Tay-Sachs to the lysosomes. So it's important that we understand that. We also have Huntington's disease, which is our autosomal dominant trait. And so you don't know that you have the neurodegenerative disease until like later on in life after you've passed on those traits. But it is a autosomal dominant. So if you have one dominant allele, you have that disease. In terms of chromosomal changes, we could see Down syndrome. And so on March 21st, 321, we celebrate Down syndrome awareness. And so that has to do with that you have 3 sets of the 21st chromosome. There's, of course, other diseases that occur with other ones, like I believe if you are trisomy 18, that's Edwards syndrome, I believe. And so there's a lot of different ones. What they could give you is this karyotype right here. The karyotype is showing you the different chromosomes that we have. And then it's having you determine what could potentially be wrong. Well, since I have 3 of these chromosomes, that is what the issue is, that they have 3 sets. This results from non-disjunction, where we don't see the 2 are not going to segregate during anaphase. And so we could end up with an extra set of chromosomes, one extra chromosome 21, which leads to Down syndrome. Klinefelter is again non-disjunction. It's XXY, so they have 2 X chromosomes and the Y chromosome. And then Turner syndrome is where they only have 1 X chromosome and they don't have a second sex chromosome. Would you ask you to calculate? I can't see what it says because of the fact that the little heart is on top of it. So hopefully someone else will comment in the chat and it will move up and I can see what question you have. So this is also where we see we can see where we have phenotypic plasticity. Okay, so phenotypic plasticity has to do with that your phenotype, your genetic material says you should have a certain phenotype, but based on the environment, you have a slightly different phenotype. And so this has to do with that the environment is affecting the gene expression. So individuals that have the exact same genotype could show slightly different phenotypes in these different environments. Okay. And so here you can see that they like cut off the skin right here, or not the skin, but the fur on the rabbit. And so usually the rabbit's feet, hair, ears, and nose are black. But if they go and they put the ice pack on there and make it cold, similar to how like the ears and the nose and all the little extremities would be, the new fur actually grows back black. So based on the environment of this cold, that causes those genes to be activated, and it then makes, of course, black fur. And you can't see that because my face is on top of it, but if you could see the picture, you would see a rabbit that has their back black. Okay, so we can also see that the diet can affect it. So here we have 2 different birds, and based on the amount of carotenoids in their food, allows you to have slightly differences in their phenotypes. So this one's more yellow, while this one is more red, because of the fact that they're eating slightly different food. And then our hydrangeas are a big example. The environment, if the ground is more acidic, they're more blue. And if the ground is more alkaline, more basic, then they're going to be pink. Okay, so you can kind of see slight differences in the phenotype of the flower. Okay, so let's talk some multiple choice and free response examples. Okay, so humans have the diploid number of 2n, or 46, which of the following statements best predicts the consequence if meiosis did not occur during gametogenesis. Okay, so again, this is just talking about how we make gametes. This is just asking about meiosis. And so in meiosis, we start out being 2n, and then we go through a rounded division, and I now have, of course, 4n, right? And so what would happen if we didn't actually go through meiosis? So if I started out with my cell being 2n, and then I have those 2n cells combining. So 2n plus 2n gives me 4n. And so that's going to show me that my chromosome number is actually going to double with every generation because of the fact that I have these 4n's. Now this 4n and this 4n mate, and they now make an 8n individual. And so like you're going to keep seeing the doubling. And so we have to make sure that we go through meiosis to ensure that I don't have double the amount of chromosomes. Okay. Here, there's a student in biology class. They crossed this Drosophila melgastar, having a gray body, long wings, with a female having black body and aparius wings. Okay, so the following distribution of traits was observed in the offspring that you can see in this data. Which of the following is supported by the data? And so here I have gray and long, which is one of my parental traits. Here I have black and Imperius, which is another one of my parent traits, right? So we have gray, long, black, Imperius, okay? And so these would be my parental traits. And then these are considered recombinant traits. And so I would expect that I would have a 1-to-1 to 1-to-1 ratio with this because of the fact that due to independent assortment and theoretically if they're on different chromosomes, I would see equal numbers of all 4 characteristics. But I don't. I see there's these ones right here, which tells me there must have been some type of crossing over that led me to have some recombinant traits. And so which of the following is supported? Gray and long is dominant. That doesn't tell me anything. Gray and long are recessive. Again, doesn't tell me anything. Genes for the 2 traits are on 2 different chromosomes and independent assortment occurred. If independent assortment occurred, I would have seen a 1-to-1 to 1-to-1 ratio. And so it must be D. Genes for the 2 traits are located close together on the same chromosome and crossing over occurred. And that's how you got these recombinant traits is due to that crossing over. So there's that. Here, I have a free response question. It's from 2016. It's number 7. And so they have this certain trait. It's 2N, so it has 4 chromosomes. The flower color is controlled by a single gene on the chromosome, which is green is dominant to purple. And the height is dwarf is dominant to tall, okay? Keeping in mind that they have told you the traits and they've given you the letters. So you must use the traits they give you and you must use the letters they give you because they've given them to you in the prompt, okay? Individuals of the parent are capital G, capital G, capital D, capital D, and all homozygous recessive, and those were crossed to produce an F1 progeny. So if I have homozygous dominant crossing with homozygous recessive, I know my offspring is going to be heterozygous. And so since that happened for both traits, my F1 is going to be heterozygous, or actually be a dihybrid. They have a heterozygous for both traits. So part A wants us to construct a diagram to depict the 4 different products of meiosis. So I'm looking for like gametes. Now they should only have half as much as genetic material. So I would expect there to only be 2 chromosomes in each of my circles, okay? And so we need to then show those chromosomes and the alleles they carry and then assume that they're on different chromosomes and the gene for flower color is on chromosome 1. So since I know there's going to be 2 on both of them, I'm just going to go ahead and just draw those on my circle, okay? I've just made a long one, which is my chromosome 1, and a short one, which is my chromosome 2. How you draw does not matter. They just need to make sure that you have 2 of them in each of them, one long, one short, okay? So due to meiosis, right, I'm going to duplicate. So there will be 2 capital G's and 2 lowercase g's, okay? And so due to the fact that we went through independent assortment, there's going to end up being 2 of them in 2 of the cells and the other 2 are in the other 2 cells, okay? So independent assortment, they separated all that good stuff. So I have 2 that have capital G and 2 that have lowercase g, okay? Now, the second allele, again, I'm going to have 2 capital D's and 2 lowercase d's, okay? But as I said, independent assortment, they can either line up together or they could line up opposite. And so because of that, I know that I need to have the 2 capital letters together, and then the lowercase letter will be with the capital letter. And the same thing with my lowercase d, okay? So I should have the 4 different options, where this one is all dominant, all recessive, and then 1 dominant, 1 recessive for each of your genes. And so that is how you would make your gametes, okay? So then part B is to predict possible phenotypes and the ratio in a test cross, which I have a certain font and it doesn't let me use numbers. So I must have forgot to do that one. Let's just say it's F1 individual. I'm sorry for that typo. And so if I did a test cross means that it's being mated with a homozygous recessive for both traits. And so if I cross this with a homozygous recessive, okay, I would expect there to be half green and half purple. And then I expect to be half dwarf and half tall, okay? Now, I'm not done because I need to figure out what the overall traits are. And so as we saw earlier, you can multiply. So if there's half green and half dwarf, that means that 1/2 times 1/2 is 1/4. So 1/4 is going to be green dwarf. And so you could do this where you say, okay, well, 1/2 is going to be, 1/4 is purple dwarf, 1/4 is purple tall, 1/4 is green dwarf, and 1/4 is green tall. And you could get that 1-to-1 to 1-to-1 ratio, okay? Now, keeping in mind, you want to write this in a complete sentence, okay? The phenotypic ratio would be 1 purple dwarf to 1 purple tall to 1 green dwarf to 1 green tall, right? Write it out, full out, okay? And so here you see the 1-to-1 to 1-to-1 ratio on the scoring guideline. So then part C says if the 2 genes were genetically linked, so if they're on the same chromosome, how would the proportions of those phenotypes most likely be different in a test cross, okay? So if they're actually on different chromosomes, right? So we already did if they were on different, right? 1-to-1 to 1-to-1. But what if they're together? If they're together, I should see that the green and the, what was it? The green and tall should be inherited together. And then I should see that the purple and, I'm sorry, I said that wrong. That the green dwarf should be together and that the purple tall should be together. Okay, those were my parents. And so I should expect there to be mostly the parental traits. I should expect most of them to be parentals. There might be some due to crossing over, but they should mostly be those parental traits. Okay, and so the majority or greater than 50% is going to have the parental phenotypes. Greater than 25% is green dwarf. Greater than 25% would be purple tall. Or the recombinants, which would be less than 25% would be green tall, and less than 25% would be purple dwarf. Okay, because those would be your recombinations, and so I would expect there to be less of that 1-to-1 ratio there. Okay, so in the 2020 CED, we also have some practice questions, and this is symbolic of number 5. So it's a short FRQ, and we're going to be annotating a diagram in some way. So in humans, the gene that determines a particular condition has only 2 alleles, one of which is capital B is completely dominant over the other, lowercase b. The phenotypes of 3 generations of a family with respect to the condition are shown in the pedigree in figure 1, and the individuals are numbered. Okay, so they've numbered them 1 through 22. Another way you might see this numbering is they might just say this like 1, 1, 1, 2, and then they would have out here these same numbering of 1 through 8, and it would be like 2, 1, 2, 2, 2, 3, and it would be like the row and then the individual number. Okay, so Part A always asks us a content question. It's always asking us about the biology. So describe the process in eukaryotes that ensures that the number of chromosomes will not double from parent to offspring when gametes fuse during fertilization. Okay, and so what they're wanting to do is describe meiosis, right? We're going to talk about how those homologous pairs are going to segregate or separate during meiosis 1, and that causes your gamete to be haploid, and then each is only going to get one member of each of those chromosome pairs. So they're wanting you to talk about meiosis here, okay? So then part B asks us to explain how chromosome from individual 16 contains DNA that came from individuals 1 and 2. So how would this individual that's so far down here actually get trait? Well, this is the granddaughter of these 2 individuals. These are the grandparents, and this is the granddaughter, right? So you can see that their father, like the 16's father is number 5, and then that's the son of the 1 and 2 cross, okay? And so during meiosis, right, number 1 first, we see the sperm and the egg, right? That's how we got 5. Okay, sperm and egg came together. So we should have a homologous pair in number 5. Okay, 1 came from individual 1 and 1 came from individual 2. Okay, so due to crossing over that may have occurred during this, there could be one of the traits, one of the recombinant chromosomes that came from the sperm is going to have both the genes of individual 1 and individual 2 on it. Okay, so you talk here, individual 5 inherited 1 member of each homologous pair from individuals 1 and 2. During gamete formation, individual 2, crossing over occurred between non-sister chromosomes in each homologous pair. Thus, each chromosome formed and passed on to individual 16 contains DNA from both 1 and 2. So that way you've completely understood how that connection happens, okay? So then part C is always going to ask you to annotate a diagram. Like last year you were doing a cladogram. The year before that you were saying where the invasive species was. And so you're going to have to modify a diagram some way. So the CED question is having us modify a pedigree. Okay, so we need to figure out what is the genotype of each of these different traits. Well, before you can do that, you need to figure out what is the mode of inheritance to be able to determine what do they each have. Okay. So if I look here, I can see that I have, where is it? So 9 and 10 are unaffected and their children are unaffected, okay? Versus here I see that 3 and 4 are unaffected and their child number 14 is affected. So that tells me it must be some type of recessive trait, okay? I also see that I have, if this was sex-linked, there's no way that 1 could pass it on to individual 5. As well as I see that there isn't a affected female here, okay? And so I'm able to know that this must be autosomal recessive. And so since individual 2 is able to have offspring that are affected and individuals that are unaffected, I know that individual 2 must be heterozygous, okay? Because they passed on the dominant trait to individuals 4, 7, and 10, and they passed on their recessive trait to individual 5. So that's how we saw individual 5 being affected, because individual 1 passed on one recessive trait and individual 2 passed on the other recessive trait. So 5 is going to be homozygous recessive, showing that, okay? So again, we see that number 4 is also heterozygous because it has an individual that is affected. So both of these must be heterozygous. And so we're seeing that recessive trait was being passed on from both 3 and 4, which gave us 14 being affected. Okay. And then here with number 8, since I've already established that the trait must be recessive, I know that this must be little b, because homozygous recessive would allow me to have that recessive trait showing out. And then here, I have one parent that is homozygous recessive, and I have another parent that we don't know what they are. We can assume that they're heterozygous. And so actually we do know, sorry, number 7, we do know is heterozygous because of the parent being homozygous recessive. And so this individual 18 must be heterozygous because of the fact that they got the recessive trait from the mom and then they got the dominant trait from the father. That allowed them to be, of course, the dominant phenotype. And then part D says, based on pedigree, explain whether the inheritance pattern of is sex-linked or autosomal and dominant or recessive, which we've already established that this is going to be recessive because individuals 3 and 4 have a trait. They pass on that trait. And then here with 7 and 8, I can see that it is sex-linked. It's autosomal because of the fact that this father would have had that faulty X chromosome, and the mother would have definitely passed on that X chromosome. And this female is affected. So there's no way that you could have had an affected female if the parents weren't both affected. And so the disease phenotype is recessive. It's autosomal, not sex-linked. It cannot be dominant because individual 3 and 4 do not have it, but their offspring 14 does. It's not sex-linked because if it was Y-linked, all male offspring of the phenotype would have the trait, and they do not. So there was that. And then on my live last week, y'all asked me to do a chi-square. And so chi-square kind of fits in well with heredity. So I figured I would pull the 2003 question, number 1B, so we can talk about a chi-square. So we're only doing part B, and we're only analyzing the chi-square. We're not doing the other parts of this. Part A, I believe, asks you to come up with, was it dominant, was it recessive? And then also, is it autosomal or is it sex-linked? We're going to find that this is sex-linked. As you see, we do have only wild-type females and only wild-type males, white-eyed males, which tells us this is sex-linked is on the X chromosome, and it's going to be recessive because the male only has 1 X chromosome versus the female that has 2. Okay, so we are going to apply that information. So using a chi-square test on F2 generation, analyze your prediction of the parental genotypes, which we already said was. So show all your work and explain the importance of your final answer. So step 1, we're going to come up with what's our null hypothesis. And now this is the Chi-5. That's by Jay Daly. I mentioned it before for y'all. I don't remember where I mentioned it. I mentioned it in an FRQ video I did before. So in the null hypothesis, you're going to tell me that the independent variable has no effect on the dependent variable. But here I'm looking at the question asked me to analyze my prediction using chi-square. So my prediction is what the parents are going to be. So I'm going to come up with my null hypothesis saying the parent genotypes are heterozygous for E, and then it's homozygous recessive for X. And so here we see capital E, lowercase e, and here we have lowercase e. So this would be my white-eyed male, and this would be my wild-type female that I would have expected to find in this, okay? So part 2 asked me to do my calculation of my chi-square value, okay? So I'm going to cross these two to figure out what my expected is, okay? So if I cross them, I get this individual that is going to be a wild-type female. I get this individual that's a wild-type male. I get this individual that is a white-eyed female, and this is a white-eyed male, okay? So I expect there to be a 1-to-1-to-1-to-1 ratio of my wild-type female, white-eyed female, wild-type male, and white-eyed male. So I have, that is definitely a typo, sorry y'all, white-eyed male. And so if I set this up, I have 100 individuals, and I expect, of course, a fourth to be each of them, so there would be 25% of each of those. The way we do this calculation is we take the total, multiply by the ratio. So 100 times 1/4 gives me 25. Now, my observed I'm getting from the data, so my F2, we saw there were 25 that were wild-type males. I see there's 31 that are wild-type females. I have 22 that are white-eyed males, and I have 24 that are white-eyed females, okay? So that, I just fill it into the chart. And I'm going to continue going. Now, I use a chart to do chi-square. That's just like the way my brain works. And so I have my observed minus expected. So 23 minus 25 gave me -2. And then you square it. So -2 squared is 4. And then you're going to take this column and divide by your expected. So 4 divided by 25. And I would do the same thing for each of them, where I get, of course, 31 minus 25 is 6, 36, and 36 over 25. 22 minus 25 is -3, gives me 9, 9 over 25. 24 minus 25 is -1, gives me 1, so 1 over 25. Now, this is back when they didn't get calculators, and so I also wanted it to be simple math for you. And so 4 plus 36 is 40, plus 9 is 49, plus 1 is 50. So 50 divided by 25 is 2.0, okay, which is our chi-square value. So what do we do once we get our chi-square value? Like, what do I do with this? So first I need to figure out my degrees of freedom. So my degrees of freedom is going to be my total classes. I've got wild type male, I've got wild type female, I've got white-eyed male, and I've got white-eyed female. Okay, so I have 4 different classes. So 4 total classes minus 1 gives me 3. So my degree of freedom is going to be 3. Okay, I then have to go into my chi-square table. Now the calculation, the equation for chi-square is given on the formula sheet, and so is the chi-square table here, but you have to know how to use it, okay? So the equation is literally like, think summation O minus E squared over E, and that's all they tell you. And so if you don't know what a summation means, which my students, I showed them a summation earlier this year and they looked at me like I was crazy. So if you've never seen a summation before, you don't know what it means and you won't know how to use that formula. Plus, students tend to make errors when they just type things into a calculator. So the chart method helps to reduce errors. Also, make sure that you keep your decimals whenever you're doing this method. I would keep 1 decimal plus whatever my final answer needs to be, by the way. Okay, so I need to find my critical value. So we're going to use a p-value of 0.5 and a degree of freedom of 3. So my critical value is 7.81. So I'm saying, okay, well, how does the calculated value, my 2, compare to my critical value, which is 7? So my table, my critical value, the value I solved, or calculated value, I'm sorry, I should have said that wrong. My calculated value is 2, and my critical value is 7.81, my table value. And so I'm going to say that it is less than, and so that tells me that I failed to reject the null, okay? Which means that my null hypothesis is right. Now I'm saying that my null is right in quotes because you can't ever prove something's right. You just can prove that it's not wrong. But there's never a way in statistics to prove something's right. You just prove, okay, well, in this case, it's not wrong. And so that tells me that my parents' genotypes are what I said they were, which was XE, X Lily, and then XEY, okay? So that was what I had for Unit 5. There's really not a lot in Unit 5, so I did want to give you a moment to ask questions. There is a big lull between when you see it and when I've actually said it, and I keep accidentally cutting the video too early and I don't have a chance to let you ask your questions. So I'm going to kill some time while y'all have a chance to ask your questions in there. If you're not following, make sure you're following my Instagram. My Instagram is APBioPenguins. I think that's kind of logical at this point. I've been doing review. I started, depending on when you watch this video, I started on February 1st and I started on February 1st every year. And I post every single day on my Instagram stories questions. And I just start on topic 1.1 and I work all the way through. Like right now I'm on 5.6. I'll post those on Sunday night for Monday. And that can give you a quick little review as you're just sitting on the bus reviewing. My TikTok, until TikTok goes away, is AP Bio Penguins, and on there I've got review videos as well as it's just kind of a way of me, telling you all about things, as well as that's where FRQ Fridays are posted. They're also posted on Instagram too. And then my AP Bio Instagram, which is where you currently are watching this video. That's where you'll find all these review videos. You'll find all of my topic TikToks from the past, and you'll find all my FRQ videos. When I finish with the FRQs, I'm going to work through every single formula on the formula sheet so that you have an example question for all of those. So I think I have killed enough time to ask your questions. So, is the chi-square number is less than 1 on the table, we accept the null. So if the value you calculate is less than the one in the table, then you are going to fail to reject the null, which you have put except in parentheses and like in quotes. So yeah, they're accepting the null. So you're saying the null is correct, but it's just more or less saying that you fail to reject that null. You don't have the data to say that it's wrong. And then finally, I can finally get to the other person's question, where they ask you to calculate map units on the exam. They might ask you to calculate map units. That formula is not on the formula sheet, as well as the formula for Percent change is not on the formula sheet. So last week, I believe in the video, I went through how to calculate the percent change, or it may have been one of my FRQ videos, because that's an important thing you need to be able to do because you did it in your lab. And so that would be your final minus your initial over your initial times 100, and that gives you your percent change. And so it's important to make sure you know how to do that. So that's all the questions that I have in there. I hope that this was helpful. Remember, if you bow paying with just success. Bye, y'all. And of course, there's a lull. So I'm going to wait a second before I end it.